This was a collaborative study among different departments from the Vall hospital, in which we aimed to identify if the cellular immune response during acute infection with SARS-CoV-2 could predict disease outcome. For that, we studied 3 different groups of patients, in which one group was outpatients with symptoms but no hospitalization, and 2 other groups were hospitalized with severe or mild disease. We studied in depth the cellular immune response, meaning the antigen-specific T cell function, their migratory patterns, and also apoptotic cell death induced by different viral proteins. Our results show that an antiviral response characterized by IL-12 secretion together with interferon gamma and degranulation, but also accompanied by IL-10 secretion, which was probably controlling inflammation, characterized patients with disease control. We also could outline which were the viral proteins instigating each response, and consequently we emphasized that next-generation vaccines should include other proteins beyond the spike. Last, because we saw that these antigen-specific T cells had homing markers that could indicate migration towards the lung, we aimed to study if these cells could establish in the lung as resident memory T cells. Resident memory T cells are a specific type of memory T cells that we can only determine in tissues and not in blood, and they establish in these tissues to offer long-term protection in case the pathogen comes back. So we had the opportunity to determine these responses in lung resections and also in their blood of patients that recover from mild or severe disease 6 to 10 months after their initial infection. Indeed, we found specific T cells with high frequency that were specific for SARS-CoV-2 viral proteins that had the phenotype of resident memory T cells. But these cells, the function and the magnitude were very different in their blood. So overall, this study indicates which are the profiles associated with disease control and which viral proteins instigate these profiles. Finally, we also indicate that future vaccines should promote not only these profiles, but also resident memory T cells that would establish in the lung and would offer long-term protection against disease. I hope you like it.